Welcome to the deep dive. Today, we're going to get into the really fascinating area of molecular testing in breast carcinoma.
Yeah, it's like getting this incredibly detailed map of a tumor, isn't it? Really showing what makes each one tick.
Exactly. And we've looked at some sources explaining how these tests are, well, changing everything. It's leading to much more personalized treatment.
That's the core mission today, really, to unpack the essentials of molecular testing and, why it's become so absolutely crucial for understanding breast cancer treatment now.
It really is a huge shift. I mean, think about it. Treatment used to be more um, standardized, maybe. A bit one size fits all.
Pretty much, but this molecular information lets doctors tailor treatment specifically for an individual's cancer. It's a game changer.
Okay, so where do we start with understanding what these tests actually tell us? What's foundational?
Well, a really fundamental piece is the hormone receptor status. So molecular tests check if the cancer cells have receptors for estrogen, that's ER, and progesterone, PR.
Right.
And this is super important because if those receptors are there, we call the cancer ER positive or PR positive. It suggests the cancer might grow in response to those hormones.
Which means hormone therapy could work, like blocking the fuel.
Exactly like blocking the fuel. These cancers are likely to respond well to hormone therapy, which aims to stop those hormones from driving the cancer's growth.
Okay, so these receptors are like specific switches on the cells.
That's a good way to put it. Switches or docking stations.
And is this common? Finding these receptors?
Oh, yes. Very common. Around 70% of breast cancers are actually hormone receptor positive.
70%, wow.
Yeah. And what's really useful is that the tests don't just say yes or no. They often give more detail, like the intensity or the percentage of cells that are positive.
Ah, so it's more nuanced. It helps gauge how likely it is to respond.
Precisely. It gives doctors a clearer picture of whether hormone therapy will be a good strategy and potentially how effective it might be.
Makes sense. Knowing how many switches are on. Okay, what's the next critical piece of the puzzle from these tests?
The next big one is HR2 status. HR2 stands for Human Epidermal Growth Factor Receptor 2.
HR2, right. I've definitely heard of that one.
Yeah, you can think of HR2 as a protein on the cell surface that when there's too much of it or it's overactive, it basically tells the cell to grow and divide really fast.
It's like stepping on the accelerator.
Exactly. So molecular testing identifies tumors that have this excess HR2 protein. We call those HR2 positive.
And these HR2 positive cancers, they have a reputation for being aggressive. But isn't this also where targeted therapies have made a huge impact?
You're absolutely right. While they can be more aggressive, they make up about 15 to 20% of cases. This is a prime example of molecular understanding leading to specific treatments.
Okay.
We now have drugs designed specifically to target that HR2 protein, kind of jamming its growth signal.
So finding the overactive part and having a specific tool to slow it down, how do they actually test for HR2 status? What are the methods?
The main ways are immunohistochemistry, or IHC, for shortened fluorescence in situ hybridization, or FISH.
IHC and FISH.
Okay. IHC basically looks at the amount of HER2 protein on the cell surface and gives a score, usually from 0 to 3 plus SAW. Think of it like counting antennas.
Antennas, okay. So 3 plus means lots of antennas.
Right. A 3 plus score generally means it's HER2 positive. If the result is borderline, say a 2 plus dot, that's where FISH often comes in.
And FISH does what differently?
FISH looks directly at the genes inside the cell, counting how many copies of the HER2 gene there are. It gives a more definitive yes or no if the IHC is ambiguous.
Got it. Okay, so we have ER, PR, and HR2.
Yeah.
These seem like the big three initial markers. What happens if a cancer has none of them?
Exactly. That leads us to the next category. Cancers that test negative for ER, negative for PR, and negative for HR2 are called triple negative breast cancer, or TNBC.
Triple negative.
Okay. And this accounts for, maybe 10 to 15% of breast cancers.
And the key thing here is treatment options are different, right?
Very different. Because TNBC lacks those 3 common targets, it won't respond to hormone therapy or those HER2 targeted drugs we just talked about.
So those keys just don't fit the locks.
Precisely. Which means the main treatment strategy for TNBC usually involves chemotherapy.
So identifying it as TNBC is crucial for avoiding treatments that just won't work.
Absolutely essential. It guides the doctors towards the most effective approach from the get-go. It shows knowing what isn't there is just as vital.
That makes complete sense. Okay, so we've covered classifying the cancer based on these markers. What about looking ahead? predicting, say, the risk of the cancer coming back. How does molecular testing help there?
Ah, yes. That's where we get into gene expression assays. These are tests that look at the activity of a whole panel of genes within the tumor.
Gene expression. So not just presence, absence, but how active certain genes are.
Exactly. You might have heard of some names like Oncotype DX, MammaPrint, EndoPredict, Precision, which is sometimes called PM50, and the breast cancer index.
Okay, yeah, some of those sound familiar. What do they tell us?
They analyze the activity levels of, maybe dozens of genes in the tumor tissue. And by looking at this broader genetic signature, they can provide a pretty refined estimate of the risk that the cancer might return, especially for early stage, hormone receptor positive, HER2 negative breast cancer.
So for that common type, ER positive, HER2 negative,
Yeah.
these tests predict future risk.
Yes, they help predict recurrence risk. Think of it like getting a more personalized risk forecast for that specific tumor.
And how does that help doctors and patients decide on treatment? Does it influence things like chemotherapy?
Absolutely. This is a major application. These tests can identify patients who actually have a very low risk of recurrence, even though they have hormone receptor positive cancer.
So they might not need chemotherapy after surgery.
That's the crucial point. For those low-risk individuals, doctors might be able to safely forego adjuvant chemotherapy, sparing them the side effects. It helps avoid overtreatment.
That's huge. Tailoring the intensity of treatment based on predicted risk.
Precisely.
Okay. Now, beyond predicting risk and the main targets like ER, PR, and HER2, molecular testing seems to be finding other more specific targets too.
Yes, definitely. The field is constantly evolving. While HER2 was a major breakthrough, we're now identifying other specific genetic alterations in cancer cells that we can target with newer drugs.
Can you give an example?
Sure. Take mutations in a gene called PIK3CA. These can occur in some advanced breast cancers.
BRCA3CA.
If a patient's tumor has this mutation, they might benefit from a drug called alpelisib, which specifically targets the pathway affected by that mutation.
So finding a specific glitch and having a drug for that glitch.
Exactly. And another major area involves BRCA genes.
Ah, yes. BRCA1 and BRCA2. We hear a lot about those in terms of inherited risk.
We do, and that's crucial. But they also have treatment implications. If someone has advanced breast cancer and they carry an inherited mutation in BRCA1 or BRCA2, there are specific drugs called PRP inhibitors like olaparib or talzoparib that can be particularly effective.
How do they work?
They exploit a kind of vulnerability in cancer cells that have those BRCA mutations, essentially leading to the cancer cell's death. It's quite targeted.
So knowing about that inherited mutation isn't just about future risk. It can directly impact treatment choices now.
Absolutely. And then there are CDK46 inhibitors, too.
Right. I've seen those mentioned. Palbociclib, ribociclib.
Yes, abemaciclib is another one. These are a really important class of drugs. They target proteins called cyclin-dependent kinases 4 and 6, which are involved in telling cells to divide.
So they put the brakes on cell division.
That's a great way to think about it. They're often used alongside hormone therapy for advanced hormone receptor positive, HER2 negative breast cancer. It's another layer of targeted attack.
It really paints a picture of, well, incredible specificity. Moving so far beyond treating breast cancer is one thing.
It really is. We're seeing it as a collection of distinct diseases at the molecular level, each potentially needing a tailored strategy. And this ties back to what you said about inherited risk. That's another key role for molecular testing, specifically germline testing.
Germline meaning inherited, right? Not just in the tumor.
Exactly. Germline testing looks for inherited mutations in genes like BRCA1, BRCA2, but also others like TP53, PTEN, and more.
And why is that done?
Well, primarily to assess the patient's overall risk of developing certain cancers and also the potential risk for their family members. It provides crucial information for screening and prevention strategies.
And as we just discussed, it can also influence treatment if cancer is already present, like the PRP inhibitors for BRCA mutations.
Correct. It connects risk assessment directly to potential treatment options.
Okay, so if we were to sort of pull this all together, molecular testing seems absolutely fundamental now.
It really is. Let's recap the key roles, right?
Yeah.
It helps classify the specific subtype of breast cancer.
Like ERPR status, HER2, TNBC.
Exactly. Then it helps predict how likely the tumor is to respond to different therapies, hormone therapy, HER2 drugs. It estimates the risk of the cancer coming back, which guides decisions about things like chemo.
Right, the prognostic tests.
Yep. It identifies specific molecular targets for newer targeted drugs like PIK3CA or using PRP inhibitors. And finally, it assesses that inherited or germline risk for the patient and their family.
Wow. That's a lot of critical information from these tests.
It really is. The bottom line is that this deeper molecular insight allows doctors to tailor treatments much more effectively.
Leading to better outcomes, hopefully, and maybe minimizing unnecessary side effects, too.
That's the goal. Personalized medicine at the molecular level, improving the standard of care.
So, I think the key takeaway for anyone listening is clear. Molecular testing isn't some niche, futuristic thing anymore. It's really a standard, essential part of modern breast cancer care.
Absolutely. It provides that vital blueprint needed for personalized and effective treatment decisions.
It really makes you think, doesn't it? If we can achieve this level of detailed understanding and tailored therapy for breast cancer, what might the future hold for other diseases? Could we see this kind of personalized, molecularly driven approach become the norm across medicine, moving away from those one-size-fits-all strategies entirely?
It's a powerful thought, isn't it? The potential implications are huge. Definitely something to ponder as this science